First of all, I would like to thank organizers for giving me opportunity to share my work on gene discovery with you. This is a snapshot of 10 years of work which we started in 2005. Harvans and I started this work in 2005 when I joined this institution in September 2005. So what is Watkins collection? It's a collection of landraces which were collected by Arthur Watkins in 1920s from 32 countries, plus there are some unknown countries as well. He used Board of Trade of London to import this material. And in beginning, there were 7,500 landraces. Those were purified and some were lost because of poor storage conditions. At present, we have 1,200 landraces. Out of these, 800 are hexaploid wheats and 400 are durums. So how they look like? These landraces, they are taller than even our students who are 170 centimeters tall. Not only variation for biotic stresses, we have variation for abiotic stresses. In addition to rust, we have seen variation for powdery mildew, for abiotic stresses as well. There is variation for grain size, root architect, physiological trait like mesophyll conductance and water use efficiency, and variation in alleles for grain hardness. So why we selected this collection? The idea behind selecting this collection was that we might have left some variation while breeding for modern wheat during incorporation of dwarfing genes. So this was a filtering process. We might have used half of the variation or even less than half of the variation. Secondly, this material was easily available and no quarantine or MTA was required because these landraces were available in Australian winter cereal collection. What we did with this material, we screened this material against stripe rust, leaf rust, stem rust in field for more than 3 years. And we did some preemptive screening. For example, for durum leaf rust races, we don't have any durum leaf rust races in Australia. Material was screened in Minnesota by Jim Colmer. Similarly, Ethiopia has durum leaf rust races which are different than US leaf rust races. And at the same time, Ethiopian races have SR13 virulence, which is not available in Australia. Material was screened in field as well as in greenhouse in Ethiopia for 2 years. We haven't left UG99 behind. Material was screened in field as well as in the greenhouse in Minnesota. There are always challenges associated with landraces, and this time the challenge was photoperiod sensitivity. Some of the landraces didn't flower even up to maturity. They were at the ground level. Therefore, we consider India. India has wide spectrum of durum leaf rust races as well as virulence and avirulence for SR13, SR24, and SR28. Therefore, material was screened in India by Dr. Bharadwaj in Shimla. After screening this material in field, we did multi-pathotype testing for all the 3 rusts and compared the result with differential and postulated genes. For example, for stem rust, we postulated SR7, SR9G, SR12, 13, 28, and 30. Lr16, 70B, 20, and Lr73 for leaf rust resistance. Similarly, for stripe rust, we had YR3, YR4, YR6, 7, and 67. Along with this named gene, we have uncharacterized gene for all the 3 rusts. For looking for adult plant resistant gene, we tested markers for known adult plant resistant gene. For example, LR34, we use CSLB34 marker to see the presence. 10% of the Watkins collection from hexaploid wheats carry LR34. 30 lines out of 800 carry Yr67 when we tested it with gene-linked SNPs. And scenario was entirely different when we used SR2 linked marker CSSR2. None of the line was positive, but we could see the SR2 linked trait, pseudo-black shaft, and seedling chlorosis. Now the question is there any other adult plant resistant gene or genes linked with PBC or seedling chlorosis? We are looking the answer for this question. We have done additional work. All the collection was screened, genotyped by 90K assay. I am presenting only hexaploid weeds here due to time constraint. What we did in this, we found 38,000 SNPs are polymorphic in 800 weeds. And we did analysis of individual data set. We found 153 SNPs significantly associated with leaf rust and scattered over 17 chromosomes. We compared the location of known genes with this association panel, and we found, for example, in this case, chromosome 6A and 5A and 6A known gene has been located from breadbeat. And similarly, 3B has Lr627, which is a complementary gene and effective when Lr31 and Lr12 is present. And we have looked into the chromosome where named genes are present. For example, alleles of Lr2, Lr15 and 22, they are located on chromosome 2DS. But we found significant association on chromosome 2DL, and we found a new gene for leaf rust, adult plant leaf rust resistance. Similarly, analysis was done for stripe rust resistance. We found 133 SNPs significantly correlated with the stripe rust resistance scattered on 18 chromosomes. The chromosomes with star like 4A, 5A, and 6A, 5D and 6A, no named gene was located. We have identified and named one gene Yr51 in chromosome 4AL. Similarly, we have new gene which will be named shortly on chromosome 6A. The analysis was done for stem rust as well. There was no named gene located on chromosome 5A and 5B before, but we identified and named gene Yr49 on chromosome 5B. And we have in pipeline genes on other chromosomes as well. So in summary, we can say when we screened this material in field and in greenhouse and did association mapping, we proved our hypothesis, which we believed in the beginning that we might have left some genetic variation behind. And we have, while identifying the new sources, we are proving that yes, there is a variation. Now, once the sources are identified, the process of characterization begins. Resistant parent is crossed with susceptible parent. F3 population is developed. Then F3 population is phenotyped in greenhouse or in field, depending upon the nature of gene. If the segregation is monogenic, life is very easy. But if we see two genes, then we are dissecting the population and isolating the single gene and developing real populations. Then DNA is extracted from the contrasting or two tails, resistant and susceptible individuals, and sent for genotyping using high throughput technologies like 90K or GBS. Once we have data back from them and linked markers were converted into PCR-based markers and tested on the whole population, and new resistant loci are identified. Sometimes we are using, to prove the uniqueness of a gene, we are using dietilo stalks or deletion stalks to prove their uniqueness. What we achieved while working on this collection, we formally named couple of genes. This is list of genes which we named in last couple of years. We started with Yr47. When we were mapping this gene mapped exactly at the same location where Yr52 was. And we looked back into the record and we found Yr52 was named by Canadian group. And when we looked at Peter Dick's work, he named this gene as LRW in 1989. And then we compared the landrace. He gave PI number and our was OS number. And it was the same line Yr63 from Iran. And these genes, they are 0.4 centimorgans apart. We have now closely linked marker. This is a result of one of our PhD students who did fine mapping, and Yr65 is closely linked to this. Sun markers, they are representing Sydney University. Yr51 and Yr57, they are isolated from one landrace which originated from Balochistan, which is now in Pakistan. Closely linked markers have been developed, and this gene is being transferred into breeding material. Similarly, Yr63 is a gene mapped in chromosome 7bs from a landrace from Portuguese. Yr72 was mapped on chromosome 2bl. We faced a number of problems in this because there are a couple of genes located in this chromosome. For example, Yr5, Yr7, Yr44, 53, 54. So we use deletion stock to prove the uniqueness of this gene, and flanking markers are available for breeders to use. Sr49 was another challenge because this gene was mapped in telomeric region of chromosome 5bl. No marker was available in the public domain, so we used survey sequences. This work was done in collaboration with B.Eth Keller. And we have flanking markers for this gene as well. So there are some more genes in the pipeline, which will be named shortly. We are confirming their uniqueness, or 1 or 2 tests are left. First 3 are for stripe-rust resistance, and these landraces are from different places. Now coming to stem rust, we are working on a couple of adult plant resistance genes, and when we are working on adult plant resistance, it takes more time than the seedling resistance genes because we can test this material only once in a year. Yr1141 is one example. We crossed this with an Australian cultivar YTP, and real population was developed. We mapped adult plant stripe rust resistant gene, which is effective against Australian pathotype as well as UG99 in chromosome 5DL, and it's not SR30. And in addition, we found a co-located QTL for leaf rust and stripe rust, which is contributed by an alternative parent YTP. So therefore, there is sometimes added advantage of alternate parent as well. Similarly, we have adult plant leaf rust resistant gene, which we isolated from 692 landrace. This landrace originally have more than 2 genes. We produced single gene lines and mapped this gene in chromosome 2DL. We are not working only on Watkins collection. We have other sources of resistance identified, characterized, and formally named. Harvans is working in this program from last 20 years with breeders, and he is going to field every day working in the greenhouse, and I think no gene can skip his eye. We have named genes from winter wheats, durum wheats, Indian cultivars, and Australian cultivars. Here I would like to give you an example of adult plant resistance gene in 2 of the Australian cultivars, Sentinel and X. These cultivars in the field produce low rust response, RMR type. And we did genetic analysis. We found 3 rust resistance genes. One is Lr46. And we tried to characterize this gene at different stages, growth stages in the greenhouse by altering temperature. And we found that genes are effective in both cultivars at 4th to 5th leaf stage at 21 degrees. And this gene in X cultivar has been identified and mapped in chromosome 7bAL, and this case has been given to Bob for formal naming. So for marker development, we are not using only 90K or GBS resources. Whatever is available in the public domain, whether markers in the physical maps or survey sequences, we are using to saturate the chromosome. This is one example of mapping of SR56, which is also located in chromosome 5BL. This was named as QTL this was identified in the beginning as a QTL from arena fauna population, and then we Mendelized by developing another population in arena GTP and saturated this region using 90K SNPs as well as survey sequences and named as SR56. This is an adult plant stem rust resistance gene like SR2. Recently, we are using another technology, that is flow cytometry. In this, we are isolating the target chromosome from resistant as well as susceptible parent, and then sequencing it using high-seq and assembling the reads, followed by marking the region using flanking markers on the zipper and then moving in towards the gene. This is done by aligning the sequences from resistant and susceptible parent and looking for indel or SNPs and designing markers around the SNP. This is example of one of the gene Yr51, which is located in chromosome 4AL, and we were having 8 recombinants in the beginning, and doing this work, we have 2 recombinants between the marker and gene. We are not leaving the genes behind. We believe in an approach from field to lab and lab to field back, and whatever we are discovering, we are deploying it in the Australian cultivars. This is one of the examples from the work done by our PhD student who was John Allwright Fellowship under ACIR program. He named 2 genes, Yr51 and Yr57, and we picked up stem rust resistance genes carefully, along with leaf rust resistance gene. We picked up 2 Australian and 2 Indian cultivars. Indian cultivars because he came under Indo-Australian project. And we combined, and donors have been developed. Seed is being multiplied and will be available to the breeders in India as well as in Australia. We are giving not only genes and donors, we are giving a package to breeder with markers as well. Human resource development, that is another mandate of our program. It's mandate as well as added advantage. Students are backbone of the program. In last 10 years, 10 students graduated from our program, and they are working in different institutions. At this moment, we have 7 students. They are really working hard. They are from different places. 1 is from Ethiopia, 2 from Uganda, 2 from Pakistan, Sri Lanka, and from Australia. They are big strength to our program. So we are also collaborating with other organizations. For example, under ACRCP, University of Sydney, CIMMYT, University of Adelaide, CSIRO, we are working together. We are screening our material in Mexico, as well as they are sending their material for screening to Sydney. We are collaborating with CSIRO heavily on map-based cloning. Sam did cloning of Yr63. He was registered with University of Sydney for his PhD. Now, recently, a new technology, RENSIC, has been developed, and we are collaborating with JIC and 2 blades, and we want to clone stripe rust resistance gene, and MTA has been signed, and we are focusing on 2 stripe rust resistant gene, Yr51 and Yr57. Mutants have been developed and characterized, and we will see soon the outcome of this cloning work. So take-home message. I can say that Watkins landraces have abundant variation for rust resistance. Several resistance genes have been identified and named, and markers have been developed. These genes are being transferred into Australian cultivars. We are targeting stripe rust resistance gene for cloning using RENSIC technology. In the end, I would like to acknowledge GRDC, ACIR, and BGRI for funding this program, and other collaborators, Matt Haddon and his team, Justin Fares, Beat Keller, Brent Wolf, Miroslav, and Evans Group for helping us in marker work, and Martin, Jojin, Jim Colmer, Sridhar, Besihun, they screen the material against different rusts. Thank you very much.